nutrition and unicellular. I'm going to take the example of amoeba. It's a unicellular aquatic organism. It's a unicellular organism where protoplasmic grade body organization is found. It's holozoic in nature. And one more thing is there, it's because it's unicellular, there is no specific system for digestion, but it will follow all these steps. The steps are, the first one is ingestion, the next one is digestion, after that absorption will be done, the next is assimilation, and after that the last one is digestion. Now I am going to draw all the diagram for this. Amoeba is a unicellular irregular that contain the irregular body shape. It's the nucleus of amoeba. In its body, protoplasm is also found. That is its protoplasm. Although it's unicellular, but it's a eukaryotic organism, so all kind of cell organelles are also found here. So you can see the specific structure, lysosome. These are helpful in the digestion. As the food particle come nearer to amoeba, it can judge the food particle. The first question arises in your mind that how the unicellular organism can judge it. It can judge it by the chemical sensation. For the receival of all the chemical sensation, the receptors are found at the cell membrane. So by the chemical sensation, it can judge there is a food particle and it want to ingest it. Now as soon as the food particle is nearer to the amoeba, it will form the finger-like projections. The next step will be, it will do effort to ingest it. I think now you can see all the efforts of amoeba to ingest the food particle. It will form the finger-like projection. These finger-like projections are also helpful in the locomotion of amoeba. These are called as pseudopodia. Just break this word. Pseudo means false, poda means feet. So these are false feet-like structures which are helpful in the movement as well as the ingestion process. After in this step you can see a small pore is also available that act as the mouth. It's formed by a cell so known as cytostome. That means cell mouth. Just by the formation of cytostome you can see a bag or sac-like structure here. This one, this is a cup-shaped structure. This is called as food cup, in which you can see the food particle here. This is the food. In the next step, it will engulf the whole food material. That's why it's holozoic in nature. You can see the pseudopodia will meet at a point. They will gradually raise its size and they will meet at a point. And now after that you can see a vacuole-like structure in which the food particle is embedded. This one is formed here, just like it. This is the food vacuole. As soon as the food vacuole is formed, this food vacuole will be surrounded by the digestive bags. These are lysosomes. Now you can see multiple lysosomes around the food vacuole. Because these are digestive bags which contain the digestive or hydrolytic enzyme. So as soon as the food vacuole is formed, lysosomes surround food vacuole, and in the next step it will get fused with the food vacuole now and form the phagosomes there. It's not growing with the ingestion process. I am drawing the large diagram so that you can see all the things very clearly. Now all these lysosomes get fused with the food vacuole and form the phagosomes. By the fusion process, they will pour their enzyme inside the food vacuole. With the help of all these hydrolytic enzymes, now the food particle will break down into the simpler and soluble substance, and that's why you can see the structure like this. When the digestion is started here. Now in the last diagram, you can see what's going on in the food vacuole. In this food vacuole, you can see the food particles are break down like this, and some undigested remains are also. Now digestion has been done. After the digestion, the next step is absorption. What is absorption? It's a diffusion of the simpler and soluble or digested food into the blood or in body tissue, body fluid, that is protoplasmia. So as soon as the food is break down into the smaller ones, smaller particles, it get diffused throughout the body. So here you can see the diffusion of food particle in the protoplasm. Now if the material is moving out of the food vacuole, its size will definitely reduce. So in the next step, I have to draw a smaller food vacuole, which is filled by only the undigested remains. Here only undigested remains is filled. And now, one more thing that you can see in my diagram, here the food vacuole is moving in the opposite direction. Why it happens so? Because in the unicellular organism or in every cell of the body that contain a vacuole, there is a movement of protoplasm which is called as cyclosis. And because of cyclosis, it's moving in the opposite direction. It gets fused with the help of cyclosis. You can see it will move towards the periphery of the amoeba, and somewhere it get diffused with the, somewhere it get fused with the body line, and now the undigested remains is removed out of the body. Here it's dumped out. As all the steps are clear, it's ingestion. Now I'm going to tell about all the steps again. The first one is ingestion. It's intake of food particle, how it can sense the food is there. By the chemical sensation, it can recognize the food particle is nearer to it, and now it will proceed for the engulfing of food material by forming the pseudopodia. Formation of pseudopodia will generate a pore-like structure that is called as cytostome, and for just beneath the cytostome you can see a cup-shaped structure in which food is held. This is called as food cup. After that, the pseudopodia will gradually raise their size and they meet at a point, and after that the line will continue and you can see the food vacuole in which the food particle is embedded. As soon as food vacuole is formed, this is first step is over now, that is ingestion. As soon as the food vacuole is formed, it's surrounded by various digestive bags. These are lysosomes in which hydrolytic digestive enzymes are filled. They get fused with the food vacuole and form phagosomes, and by this process they will dump their enzyme into the food vacuole where food particle is found, and it's directly digested into the simpler and soluble form. Its digestion, this chemical digestion is over, and in the next step the chemically digested food will diffuse towards the protoplasm. That is the third step called as absorption. Now this food is absorbed in the protoplasm, and this amoeba will utilize the food material in the movement, in the formation of various substances, to get energy, to do various metabolism. So you cannot draw the assimilation. Assimilation cannot be drawn because it's a process of where the processes are going on. You cannot show it by diagram. So you have to skip this step in the diagram, and the last step is ingestion, in which you can see the food vacuole is smaller here because of the diffusion of substance, and now it will move towards the opposite end. That was the entry point, and that will move towards the opposite point where it get fused with the cell membrane and the undigested remain is adjusted out. For the digestion process, anus is formed. That anus is formed by a cell. That's why it's called as cell anus or known as cytopage. It's called as cell anus. With the help of cytopage or cell anus, this undigested food is expelled out of the body. It's over about the nutrition in unicellular organism. Thank you all.